Hi everyone, I'm Megha and today I'll be presenting the topic gene mapping by interrupted mating in bacteria. So, these are the topics that we'll be discussing in this presentation: gene mapping, interrupted mapping, bacterial conjugation, the properties of F plasmid, recombination between donor and recipient DNA, mapping by interrupted conjugation, F factors carrying bacterial genes. So let's move on with our presentation. Gene mapping. Maps present information about the world in a simple visual way. It shows the location of different places, the distance between two locations, etc. Similarly, gene mapping is the process of establishing the locations of genes on the chromosomes. Earlier methods of gene mapping used linkage analysis, and by this, closer the two genes are to each other on the chromosome, more likely it is that they will be inherited together. So by studying the inheritance patterns, the positions of genes can be determined. However, more recently, scientists have been using recombinant DNA techniques to establish the actual physical locations of genes on the chromosome. What is interrupted mating? It is actually a technique that is used to map the bacterial genes by determining the sequence in which the donor genes enter the recipient cells. And this sequence in which the donor genes enter the recipient cells is determined by disrupting the bacterial conjugation after specified intervals of time. We have already learnt about bacterial conjugation. However, we'll just refresh our memory regarding that topic. So what is bacterial conjugation? Conjugation is merely the fusion of two compatible bacterial cells. And conjugation and gene transfer can only occur if there is a circular plasmid or circular DNA plasmid called the fertility factor or the F factor or the sex factor, which is found in some cells but not necessarily in all the cells. So only if there is an F factor can conjugation and gene transfer occur. And the cells that carry this F plasmid are called or designated as the F plus, and those lacking it are called F minus. And the plasmid, F plasmid, contains about 100 genes. Coming on to the properties of F plasmid. F plasmid can replicate its own DNA. And the cells that carry this F plasmid promote the synthesis of pilus on the bacterial cell surface. What is a pilus or pili? Pilus is a hair-like appendage found on the surface of many bacteria. This is an appendage that is very much important for bacterial conjugation. Their function include mediation of cell-to-cell interactions, motility, and DNA uptake. Then, the conjugation can take place between F plus and F minus cells. F plus cells are inhibited from making contact with other F plus cells. So the F plasmid cannot be transferred from one F plus cell to another F plus cell. That transfer can only take place between an F plus cell and an F minus cell. Sometimes this F plasmid carries one or more insertion sequence elements. And it is using this insertion sequence elements that the F plasmid or this F factor integrates with the genome of the bacteria to form an HFR cell or high frequency recombination cell. So high frequency recombination cell is actually a bacterium with a conjugative plasmid or the F factor integrated into the chromosomal DNA. This integration is usually by homologous recombination. The integrated F factor in the HFR cell sometimes leaves the chromosome and moves back into the cytoplasm. And while doing that, it might carry some of the host chromosomal gene along with it. And this modified F factor is now called an F prime. And this F prime can now transfer that host specific genes to a recipient cell during normal conjugation. So you can see here in this picture, F factor can replicate its own DNA. And here you can see F factor promotes pilus formation, which promote cell-to-cell contact. And you can see here F plus cell can contact or come in contact with an F minus, not another F plus. And you can see F is being transmitted or transferred into the recipient cell. And a copy of the F remains in the donor cell. And here you can see an insertion sequence elements, and as I told you, it is with that insertion sequence it gets integrated into the bacterial chromosome and forms an HFR cell. You can see here the F factor with the insertion sequence element get integrated to the chromosomal DNA. Now it forms an HFR cell. Now sometimes normally it gets transmitted to another recipient cell, but sometimes it may move away from this chromosome, this integrated form, and move back into the cytoplasm carrying some of the host genes. And during another conjugation, this host specific genes can be transmitted or transferred into a recipient cell. You can see two bacterial cells here. This is one bacterial cell, the other one is another bacterial cell, and you can see an F factor. So the first one which carries an F factor is called an F plus cell, and the other one which does not carry that F factor is called as an F minus cell. And as I told you, only F plus cell and F minus cell can conjugate. Now the donor cell carrying the F plasmid or the F factor forms a structure called as the pilus that connects with the recipient cell. And to start the conjugation, the F factor is cut at a sequence called as the origin of transfer by a protein assembly called as the relaxosome. This complex associates with the strand to be transferred on the tDNA. The accessory proteins of the relaxosome are released while a protein called as the relaxase remain attached to the tDNA. This relaxase tDNA complex is recognized by a coupling factor and transferred to the exporter, which is a complex of the F plus cell which is continuous with the pilus. Now this exporter pumps the relaxase tDNA complex into the recipient cell. Once the relaxase tDNA complex is inside the recipient cell, the relaxase causes the circularization of the tDNA. When the tDNA of the recipient circularizes, the tDNA gets replicated and forms a double-stranded structure in both the cells, both in the recipient cell as well as in the donor cell. At the end, the cells are left with complete double-stranded copies of F plasmid or F factor. The connection through the pilus will be released at the end of the conjugation, and each becomes an F plus cell. The cells can conjugate with other F minus cells. So the conjugation allows genes from two different parental cells to come together in the same cell and provides an opportunity for recombination to occur. So because of this recombination, mapping analysis is possible. So all the crosses are by definition of the type HFR cross F minus. HFR is the donor and F minus is the recipient. So after cell union, the HFR chromosome replicates in a peculiar manner that reels out a single-stranded DNA molecule which is then transferred linearly into the F minus cell. The replication and transfer begins at a specific point at one side of the integrated F factor on the HFR cell called as the origin. So the replication and transfer begins at origin, and the genes which are close to the origin will be transferred first, and the genes that are located farthest from the origin will be transferred later. The integrated F factor would be transferred last. Okay, but in most of the cases this F factor will never be allowed to transfer into the recipient cell, or it will never enter the recipient cell, and this transfer process will stop before this F enters. Once inside the F minus cell, the linear single-stranded DNA molecule acts as a polymerization template, and it will be converted into a DNA double helix. That is, it will become double-stranded. And this linear donor fragment of DNA is the exogenote, and the resident F minus chromosome is the endogenote. You can see here, this is an HFR cell, and the transfer begins from the origin. This is the origin which is near to the integrated F factor. So the origin enters first, and the genes which are closer to the origin enters earlier, and the genes which is far away from the origin enters later. And this is an X conjugate, the cell with a linear DNA and its own DNA. So this is an X conjugate. And here you can see the single-stranded DNA that entered into the recipient cell is converted into a double-stranded structure because it can replicate itself. It can act as a template for polymerization. So it is converted into double strand, and the strand of DNA that entered into the recipient cell is called as the exogenote, and the cell's own chromosomal DNA is called as the endogenote, and recombination takes place between these two strands, or the chromosome and the exogenote, and recombination takes place. You can see an F plus cell here which carries an F factor, and a cell becomes an HFR cell when the F plasmid gets integrated into the genome of the bacteria. And when this HFR cell mates with an F minus cell, one strand of the DNA is cut at the F origin sequence. The free 5' end moves through the transfer pore while the chromosome replicates inside the HFR cell. The donor strand is also replicated as it enters the recipient. When the cells separate, the donor cell's chromosome is restored in the recipient. Integration may lead to recombination of gene alleles. The linear DNA is eventually degraded. When many HFR and F minus bacteria are mixed, conjugating pairs can form quickly. If origins are nicked and donor strands begin to move into recipients about the same time in all established pairs. The donor genes closest to the leading strand of the F origins are transferred early, and the donor genes residing farther from the origin are transferred later. At any time, the mating can be interrupted. If the F minus recipient is drug resistant, the drug treatment can eliminate the HFR bacteria. Then we can check for F minus recombinants among survivors. This kind of nature for HFR mating can be used to determine the gene order in the donor chromosome.